So Ard Louis is going to kick off then this afternoon for us. He's actually a Royal Society University Research Fellow here in the University of Cambridge in the Department of Chemistry. Ard is leading an interdisciplinary research group studying the sort of problems that you get on the boundaries between chemistry and biology and physics. He's a fellow of Hughes Hall. He's currently director there of the studies in natural sciences. And Ard has an interesting background. He was actually born in the Netherlands. He was raised in Gabon in Africa, and he received his PhD in theoretical physics from Cornell University in the United States. Now, it's sad to report from our point of view here in Cambridge that soon Ard is going off to the other place, as we call it here in Cambridge. He's going off to Oxford, where he's taking up a lectureship there in the Department of Physics, but Cambridge's loss will be Oxford's gain. But we're delighted that Ard is here with us this afternoon. He's going to talk to us about the origin of life. Thank you, Ard.
Well, thanks for all showing up on this afternoon. It's always a bit of a challenge to be in the post-lunch slot. It's usually time when I fall asleep, so I'll try not to and hope that you don't fall asleep too much. I was asked to tell you about the origin of life, and so I'll try to tell you what I think about the origin of life. Slowly. Okay, sorry. Also about the origin of life. Yeah, stop me anytime. I will speak very quickly if left to my own devices. That's fine, yeah. So, one of the things you'll notice through my talk is that the origin of life is the kind of field which attracts people of a particular type, I should say. In Dutch we have a word called grootheidswaanzin, which means you think that you're, I'm not really sure what the English word is, megalomania, maybe something like it. So I'm a little bit wondering why the organizers asked me to speak about this, but there you go. I have been working on a project with the Templeton Foundation on the emergence of biological complexity. So I've read a lot of proposals on the origin of life. It's given me a bit of a snapshot of what the latest, greatest is. So I hope to reflect a little bit of that. But I hope also to steer it a little bit in the direction that you would like me to. So if you have some questions in the middle of it, then I'm happy to take a digression.
Better not. Keep it.
Oh.
Just from the film.
Oh, I'm sorry. That's okay. So
Yeah.
you can do digression at the end. Okay. So the first thing, just to put everything into focus, is to ask ourselves, where are we? What are we going to talk about? So there's a little thing here on the history of life on Earth. I've got here with the oldest prokaryotic fossils. Those are the oldest fossils we think of bacteria. Then at some point around 550, 600 million years ago, the Cambrian explosion, animals first appeared. And a lot of what you read about evolution is essentially post this. It's very interesting. There's all these mass extinctions, like this one right here at the end of the Permian, when approximated about 90% of all species died out. And there's been a whole series of around here was the KT event, when the dinosaurs died out, and us mammals were no longer stuck as little furry animals in little holes. We could climb out and become who we are now. And eventually we end up with humans. And in fact, one of the interesting things is that if you try to put this into an idea of time, then the part of the Earth time I'm going to talk about is this first bit, which is, let's say if you made this into a 24-hour day, then the bit that I'm going to talk about would be the first 2 hours of that day. Interestingly, if you think about when humans didn't really appear on this one until about the last 1/10 of a second, if you were to take all of the Earth's history into a 24-hour day. So we're really at the very last bit. And in fact, if you look at recorded history, then you're really looking at the last millisecond of a 24-hour day. It's a very, very short period of time indeed. So I'm looking at something which happened very, very long ago, if you think about it. And the part that I'll be talking about is something that happened after this, which is quite a nasty, PowerPoint. It's called the late heavy bombardment, which, so as you know, the Earth was formed from an accretion disk around the sun. The pieces of matter coalesced together and self-assembled, if you wish, into what we call the Earth. And at some point around then, there was this enormous shower of meteorites that hit the Earth. We see the pockmarks still on the moon. And those bombardments, some of them which would have been several kilometers in diameter, almost certainly if any of those things hit the Earth, if any life was there, if there had been any life, it would have been killed, it would have been vaporized. The seas would have been vaporized, everything would have gone. So at some point that finished, luckily for us, and for life. And in fact, one of the really interesting things is that the very first evidences for fossilized life, so it's not fossils, but you can look in rocks, you can find evidences in the chemistry of there having been life. The first of those are, this is quite controversial, but between 3.8 and 3.5 billion years ago. And so one of the really kind of striking things is just how rapidly life really emerged. And for a very long time, the Earth was covered mainly with bacteria, okay, single-celled prokaryotic organisms that is without a nucleus on the inside. And I have a little interlude here about bacteria, partially because it was first seen by a countryman of mine, Anton van Leeuwenhoek. Here you see a typical example. It's basically a little bag with little proteins. Those are the molecules floating around. It's got some DNA in the center which tells it how to copy itself. It's got little ribosomes here. I'll get back to them later. Ribosomes are the object which make the proteins, so they make the proteins that are essentially the molecular workhorses of the cell. You may not realize this, but in your own body, in your gut, you've got 10 times as many bacteria as you have cells in your entire body. Okay, that's not a bad thing. They're helping you digest. They're mainly inside your intestines. But that's just something to think. They're found everywhere. They're found, people have drilled several kilometers into the earth, and they find bacteria which live very, very deep in the earth. Bacteria are found in really extraordinary wide variety of locales. One thing I found very interesting myself is extremophiles. That's just a word for bacteria, or really, they're really archaea mainly, that live in extreme conditions. Like some of them live in geysers. It's called thermophilic bacteria. Thermophilic, they like heat. And they can be found in temperatures up to 115, 120 degrees Celsius, so above boiling, because these things are under high pressure. They can live in very, very high salt concentration or very high, very, very low pH, something that if you put your hand in, it would burn you, and they can live in there. They're found in these black smokers at the bottom of the sea, which are extremely hot and high pressure. They're also even found at minus 15 degrees Celsius, frozen in the ice of Antarctica. So one of the really amazing things about bacteria is that they kind of rule the earth, if you wish, and they have done that for a long time. Even now, there's about 150 times as much biomass made of bacteria as there is, for example, human biomass. So that's a bit of a guess. We don't know how much there is because there might be huge amounts of it under the earth that we don't know about, but that's a conservative guess. So they're everywhere. I think that's quite an interesting thing to think about. And when you look again at the origin of life, you realize that for most of the Earth's history, we're basically looking at microbes. They've been here for most of the time, and we've only been here for a very small percentage of that time. And a lot of what you heard of earlier, if you were here last week, some of the talks about evolution, what you may have read about evolution, really focuses on the more recent evolution. And if you go back, you've got microbes that were here for a very long time. And I'm going to talk about this bit right here. After the late bombardment, when the Earth kept being sterilized, until what seems to be the very first fossils of microbial life. During that very short period, somehow, life emerged on Earth. Now, one question you may ask yourself is, what is life? Okay, and you can ask that in lots of different ways. There's a famous quote by a judge in the US. He said, "I know it when I see it." He was speaking in this case of whether something was pornographic or art. And he said, he knows it when you see it. So there's a sense of what life is. You know it when you see it. You will know life. We recognize it. There was recently a conference on astrobiology, and a famous astrobiologist, Gerald Joyce, famous RNA world expert from Scripps, kind of came up with this definition, which everyone agreed with at that point, which is life is a self-sustaining chemical system capable of undergoing Darwinian evolution. Or you might say life is something which has both replication, it can copy itself, and metabolism, it can somehow obtain energy from the environment. Interesting, there's a recent very nice paper by Christopher Chiba and Carol Cleland. Chiba's a well-known astrobiologist and Cleland is a well-known philosopher of biology, and they make the argument that, in some sense, our difficulties in defining life in a very precise way reflect the difficulties people had in defining what water is in the 19th century, before we really had a clear idea of the molecular nature of water. And the reason for that was simply because they didn't understand exactly how water works. And so you can try to define water and say, well, water is something which is clear, it can boil, it can freeze, but it's all a little bit fuzzy and vague. And it's much clearer when you've gotten a microscopic picture. And their argument is that in some sense we're not able to define life yet because we don't have a really unambiguous definition or understanding of how it works. And people are more or less working, I think, in two different directions to understand life. One, which I find particularly interesting, is kind of a top-down picture. So you take what you know which is alive and you try to make it simpler and simpler. And a very famous example is this little organism, Mycoplasma genitalium. Some people argue it's not quite alive because it needs to be in an environment where it takes certain particular metabolites. But it has 517 genes, which is more or less the smallest genome of any known organism. So you remember, roughly speaking, each gene codes for a protein. That's the kind of classic central dogma of biology. It's a bit more complicated in practice, but roughly speaking, each gene codes for a protein. So this thing needs, on that order of that, so slightly more proteins to survive. And what people have done is they've knocked out various genes. They knock the gene out and they make the organism and see if it still lives. And they've done that all the way until it dies. It turns out that somewhere between 265 and 350. 350 because that's the least they've been able to do without killing it. 265 because there's roughly 100 other genes for which they don't know what it does. And so they're saying, well, maybe if we could knock them out. Okay. And this is an interesting way of looking at it. In fact, Craig Venter, who you may remember from the Human Genome Project, he's the chap who had the privately funded attempt to decode the human genome, which was in direct competition with the publicly funded initiatives. And he is somebody with with a well-developed sense of self-esteem, but also somebody who is very, very, very creative. And so he's now switched from working on the genome, and he's going to try to make synthetic life. The idea is he's going to try to work top-down until he can figure out the simplest possible life. And then he hopes to somehow make new versions of life that would be useful, for example, for making petroleum, making oil out of simpler substances, etc. So that's one way of trying to define life. The other way would be from bottom up. And this is the way that I'll be talking about mainly today. And this goes back all the way to Darwin, who wrote the following letter to Joseph Dalton Hooker. If, and oh, what's a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc., present, that a protein compound was chemically formed, ready to undergo still more complex changes. So this is a famous example of the warm little pond that Darwin thought that perhaps life had emerged in initially. And he pointed out as well that if that happened a long time ago, it could have sustained itself into fully blown life as we see it today. Conversely, if that were to happen today, all the microbes floating around would almost certainly eat up whatever is made. So it's not something that we're going to see happening again and again. It's something that would have happened once, perhaps not so long after the late bombardments. And that was something he never actually published openly. It was just in a letter that he wrote to one of his close associates. And there wasn't that much work on that scientifically, really, until the Russian scientist Alexander Oparin, and independently, the very famous British theorist Haldane came up with the idea of a primordial soup, which is the idea that there are these chemicals which are floating around in water of some kind, and they self-organize into some kind of duplicating system. And they come up with a few suggestions of how that might work. And this is really where the idea of primordial soup came from. Got a little picture of it here. Lightning striking down, chemicals floating around, and this mixture of energy and chemicals would somehow come together and make life. This didn't really pick up very much, certainly not in the public imagination, until a very famous experiment by Stanley Miller. Stanley Miller was a PhD student at University of Chicago, was actually working on something in nuclear science, got bored of it, and bugged Harold Urey, who was a very famous Nobel Prize winner in Chicago, to try to do an experiment working on the origin of life. And Urey said, okay, fine, you can do it, but I'll give you 1 year to succeed. And so Miller started working on this, and he came up with this little contraption here, which has an atmosphere, if you wish. It has chemicals coming in, which is ammonia and methane and hydrogen and water. It goes into it evaporates up, and in this chamber here, you've got little sparks that get shot into this atmosphere. And he got this to work, and to his great surprise, after waiting for about a week, he found a little bit of gloop at the bottom, and he looked at it carefully, analyzed it using paper chromatography, and he found glycine, which is one of the simplest amino acids. And that was an enormous surprise to everyone. In fact, Urey, to his credit, allowed Miller to write the paper on his own. And so Miller published the first paper by himself, which is quite something for a 23-year-old to do. And that was a big surprise, I think, to everyone. I don't think people had expected that something as simple as that, just electric discharge with a few simple basic molecules, would form something as complicated as an amino acid. So you remember that amino acids are the objects from which proteins are made, so they're the units of proteins. And so that was a big surprise. And later, as they tried different combinations of gases and different, in fact, this has become an enormous industry, and Miller has been leading this for quite a long time. He's, again, a very strong personality. And they found the basic, if you think of the main chemicals you need for life, so you need sugars, you need nucleic acids, which are the things that make up DNA and RNA. You need amino acids to make proteins, and you need hydrocarbons for the lipids, which are the things that make the little outside shell of, say, bacteria or of your cells. And they were able to make at least not all, at least some of the components for each of those major types of organic molecules. So that was very striking. It actually turns out that this has actually all been done previously by a German chemist in about 1913 called Walter Loeb. And I have an interesting quote here for those of you that are interested in the history of science. I think this is a marvelous piece of history to look at, just because of all the personalities interacting with each other and there's all this megalomania going around. But Arrhenius said the following, Gustav Arrhenius, that the reverence accorded to the American work is an American myth originally based on cultural and linguistic ignorance, later on unwillingness to acknowledge the original work. Okay, so perhaps it wasn't Miller who did it first, but certainly Miller was the one who popularized it and who created a field of people looking at these types of experiments. Now, Miller is still active today. There's a lot of problems with these experiments. One of the main ones is simply the atmosphere that Miller thought a reducing atmosphere is almost certainly not what people think is the atmosphere of the early Earth. And the other issues with it is that it's very hard to make some very critical components, particularly certain a sugar like ribose or some of the nucleotides like adenine or guanine. And the other thing is that when you do this kind of experiments, what you really get out is tar. So you can say, well, you can look through the tar and you can find all these interesting molecules, but you find a very wide array of other molecules as well, and they're all kind of mixed together in a fairly messy way. And those are the kinds of problems that there are with this type of experiments. Now, I have a quote here from Leslie Orgel, who's somebody I respect very much. In fact, he wasn't one of my predecessors in Cambridge. He was a reader in theoretical chemistry before he went into the origin of life type of things. And he says the following: does this tell you something? These experiments actually tell us something or not? And Orgel says, it's hard to believe that the ease with which sugars, amino acids, purines, and pyrimidines are formed under reducing atmospheric conditions is either a coincidence or a false clue patented by a malicious creator. So this is, I think, a very general feeling in the field, that even though these experiments don't quite work, it's still so surprising that you could make all these things, that must be a clue of some kind. A counter argument is this is Gunther I cannot pronounce the name, Wachtershauser. As far as I'm concerned, the soup theory is more a myth than a theory because it doesn't explain anything. So this is, again, another strong personality we'll encounter later. But roughly speaking, that's the way people feel, and probably the majority of people in this origin of life field follow Orgel, think it's somehow something really, really amazing. So that's the very first bit if you want to make life. You've got to somehow make the biomolecules from which life is made. You've got to create them in some way or another. And are these components easy to make? So this is a recent quote from Robert Hazen, who's a geophysicist who works on the origin of life. He says, "Anywhere energy and simple carbon-rich molecules are found together, a suite of interesting organic molecules is sure to emerge." And in fact, the Stanley Miller, I call it the empire because he's got a very wide influence. Lots and lots of molecules are made by experiments of that type. People like Hazen make experiments and make molecules, biomolecules under high pressure. You can make them from hot rocks, clays. You can make them from giant impacts. People study the impact of rocks very hard into each other, or basic chemicals into each other, and you can make these in the lab, and some people think that might be how we made biomolecules. They can come from deep space. Comets are typically encrusted, people think, with some kind of tar. In fact, glycine, again, the simplest amino acid, was recently found by spectroscopy in deep space by a Taiwanese group. So that's all quite striking, I think. Although I think the space bit, this is Stanley Miller, organics from outer space, that's garbage. It really is. Again, one of the interesting characters in this field. So these are the kind of different ways where molecules come from. Miller thinks this is still the best way to make them. But there are lots and lots of alternative ways of making molecules. And it's quite clear that at least the components are relatively easy to make. And that was initially a surprise, but now it's something which is well established. The other question is, can you make biopolymers? Because, of course, proteins are not just little amino acids. They're strings of lots of amino acids all stuck together. So each one connects to the other. And that turns out to be much, much more difficult. Moreover, life depends on not a very large number of organic molecules, but actually, considering the number of organic molecules that there are, life actually uses a very, very small subset of those molecules. If you think of small organic molecules, it only uses a few hundred, where there's probably something on the order of, who knows, 10 to the power 50 or 60 different simple organic molecules that you could potentially make. And the problem with all the experiments is that they all make a very large number of different ones. For example, typically your body only really uses 2 kinds of sugars, but experiments will make hundreds of different kinds. And that's one problem that you have. If you want to make a biopolymer of some kind, you want to make something that connects together, you've got to somehow separate out the components that you need. There are other very difficult problems. One of them is chirality. So this is the idea that a lot of molecules have a left or a right chiral symmetry. Here's just a little picture of them. This is glycine, again, a little amino acid, or it's actually just a little amino acid with some kind of side group on it, which has a left or a right symmetry. So, for example, there's a very famous example which you may have had in school. So, limonene, if it's the right turning type, it has an orangey smell. And if it's the left turning type, it has a lemony smell. It's found in pine needles, for example. So that's a fairly important difference. At the moment, any kind of drug enforcement, government drug enforcement agency like the FDA in the US, they insist that any drugs that are made are very carefully done in the right chiral way. This is partially because of thalidomide. You may remember this in the 1950s or 60s, I think it was, when it was used as a cure for morning sickness. And the right chiral one cures morning sickness, whereas the left chiral one induces birth defects. And this is a famous example of the subtlety with which this chirality affects life. The problem is almost every single synthesis that you can make in this type of, in any kind of origin of life type synthesis, will make a mixture of roughly 50-50. And there's a huge amount of work, people trying to find ways of making, separating out a little bit more of the right, or a little bit more of the left-handed type. But if you look at almost all the experiments, they're very different. They have very small yields. You can get a very good publication of getting less than 1% more of one or the other. And that would make you very famous. And that's still a long ways from making it purely one or the other. So that's really a big problem. Again, it's not clear how to solve that. That's a problem in making the components for biomolecules, or making polymers. In fact, probably here I've got a typical polymer, RNA, which we'll look at a little bit later. It's made of all these nucleic acids all connected together in a row. And even Stanley Miller, the ebullient Stanley Miller, says the first step, making the monomers, that's easy. We understand it pretty well, or he thinks he understands it pretty well. But then you have to make the first self-replicating polymers, and nobody knows how it's done. And that's probably the state of the art of the field at the moment. There's lots of difficulties in how to make these polymers, lots of ideas about how it could be done, and lots of tantalizing clues, but nothing that seems anywhere as successful as, say, the kind of origin of simple biomolecular components. Another thing to think about in these self-replicating molecules, which are molecules that somehow copy themselves, because that's a very basic problem for life, is what came first, the chicken or the egg? Okay, and the problem is really what came first, metabolism or replication. So metabolism is just a fancy word for the idea that you need to get energy from your environment if you want to live. Okay, and replication says that if you want to live, you need to copy yourself. Eventually you'll die, and you need to have copies, and if you make lots of copies of yourself, that's better than less copies. So that's the basic question. And the question is, which of them came first? You may know that in basic life, it uses proteins for metabolism, because proteins help catalyze reaction. That's a fancy way of saying it helps reactions to happen. On the other hand, you need DNA and RNA to replicate. And the DNA codes for the proteins, but the proteins help make the DNA and the RNA. So the question is, which one of the two came first? And you'll find that there are, in this ideas of self-replicating molecules and etc., there tends to be a split between people who think metabolism came first, some kind of proto-metabolism with imperfect replication, which then later was optimized to what we now see with DNA and RNA, and people who think that somehow some kind of replication happened first, something which copied itself, and then it developed a full-fledged metabolism. One of the ideas related to this, and this is particularly true for a lot of people looking at from metabolism first, I've got a few well-known names here, is the idea that you'll have some kind of chemical evolution is the word that's often used. Well, it's not entirely clear whether Darwinian evolution would work on this level, but it might work as a optimization strategy, and that you would have something like this. So A and B with a catalyst E. So catalyst is something which helps the chemical reaction to happen. So A plus B, E comes in, it makes C with D. G helps make E again, which connects with F, which makes B again, which uses B to make G, which then copies itself, makes C and D, and then it copies itself in that way. It's called an autocatalytic cycle, because you've got a series of reactions that happen, and the end point then starts the beginning again. And if you do that well, you can multiply yourself and copy yourself. And a lot of the ideas are that life first started in this way. Here's a particularly interesting one by Gunther Wachtershauser. This is a quite interesting chap, because he was a German patent attorney. So he wasn't an active scientist, although he did have a PhD in chemistry. And in the evening, he studied this. And at some point, he published this paper on his idea of basically the origin of life, which is based on these autocatalytic cycles. He said that these things happen on pyrite, which is fool's gold, basically. That's redox reactions, certain kind of reactions give the energy, and you get these flat life autocatalytic things happening on the surfaces of these minerals. And he's quite an interesting character, because he was close friends with Karl Popper, the philosopher. And Popper is the one who helped, in some sense, popularize his theory. Those of you that are scientists may realize this, but those of you may not realize that if you're a scientist, there is also a reasonable community of people on the fringe of science, and they can sometimes be patent attorneys or people of that nature who will have crazy ideas which they then send to you. So I regularly get emails from people of various kinds who send me crazy ideas about whatever, the origin of life or quantum mechanics or these kinds of things. And they're sometimes very clever ideas, but when I say they're slightly crazy, it's because they've been more or less thought out by themselves in the evening outside of work time, and they haven't been tested, they haven't been they haven't really interacted enough with other people to see what was right and what's wrong. Now, there are, of course, very famous counterexamples. There's a young man in 1905 who was a patent attorney, of course, Albert Einstein. So there's nothing, you may be wrong. And Wachtershauser basically was friends with Popper, and Popper was the one who initially popularized, or helped him popularize it. And like Wachtershauser said, he was very much influenced by Popper, the idea of falsifiability, which I'm sure you've heard already this week, which is the idea that any scientific theory should be falsifiable. You should be able to make a prediction which you could prove wrong. Otherwise, it's not a real scientific theory. And he felt that previous theories of the origin of life weren't falsifiable. They were kind of just so stories. So he was going to make something which was falsifiable. And so he has these amazing papers full of all kinds of chemical, very, very complicated chemical schemes, which are very well falsifiable because you can try them. Turns out they're harder to try in practice than you think. And people that do, or try to do similar things, sometimes they have this reaction. This is what he said to Morowitz, another famous origin of life person, who wrote a paper that Wachtschau didn't like, and he said, retract it or I'll sue you. This is not, science can be very competitive, but we don't tend to sue each other about these kinds of things. So he's an interesting character again. And some of these ideas have actually, some of these reaction cycles have actually been tried in experiments, and they do yield some interesting outcomes, but it tends to be very, very small, much less than 1% of the things that you're hoping to make. In fact, this iron sulfide world idea has also got people thinking about life at these deep vents. So another way of thinking, another very popular idea is that life somehow started in one of these extreme conditions. Like these are these black smokers under the, very deep in the ocean. They're very, very hot. They spew out a set of chemicals. And the idea is that they tend to have a lot of sulfur there, and that some similar reactions to the Wachtenschauer reactions could be happening there under high pressure. And people have tried to emulate some of this in the laboratory, particularly Hazen, this chap I was mentioning earlier. And they do find that this high pressure tends to generate a very interesting, rich synthesis of biomolecules. Again, the problems with this is that these organic molecules are also very unstable at the high temperatures, especially nucleotides, things you need for RNA and DNA, and they break up again. And so this is why Stanley Miller says the vent hypothesis is a real loser. And so those are just some of the ideas that are floating around. These are ideas to have more complicated biomolecules. And there's a large number of different schemes along these lines. They're all very interesting. They all, but none of them really seem to work very, very well. And the best way to figure out what's wrong with them is just to see what the people who have alternative theories say, like Miller here. Another aspect to think about in the origin of life is you don't just need to make biopolymers. You've also got to encapsulate them in something. Okay, they have to be inside something so that they can eventually happily react on their own and then hopefully copy themselves. And here's it's quite a nice proposal by one of my colleagues, Chris Dobson, which is the idea of aerosols. At the surface of water, you can, when you've got lots of wind, and you have lipids, which are the little molecules here, you can get little aerosol bubbles, which some atmospheric scientists discovered are floating around in the air as we speak. And they kind of float around, they come back down again, they float around, come back down again. And they come up with nice schemes of chemical reactions which happen in these little aerosol bubbles. The aerosol bubbles go up, they float around, there's enough time for various syntheses to happen, they come down again. And if one is particularly successful, it might become the origin, the proto-life, the very early ideas of very early versions of cells. And again, these lipids are quite easy to make by a wide variety of various prebiotic chemistry, the prebiotic schemes. And this is again one of many, many proposals that's out there, but one that I think is quite interesting. Other ideas that there exist are, you may have heard of the clay world by Graham Cairn Smith. It's a very creative idea where he believes that the very first replicators were not actually biomolecules at all, but pieces of clay which could copy themselves. And that eventually the biomolecules stuck onto them, and then they took over the replication. So that's Cairn Smith's idea. Another very interesting idea by Thomas Golds, a very famous Austrian astrophysicist, together with Hoyle, was one of the big proponents of the steady state theory of the universe, the kind of anti-Big Bang ideas. And he made himself famous or infamous with his ideas of the deep, hot biosphere, that when he realized that there are all these molecules, these bacteria very deep in the earth, living in rock almost, as it were, he thought, well, that's where the origin of life might have come out of. And so he had a whole scheme about how the origin of life actually emerged deep in the rocks. He even convinced I think it was the Swedish national oil company to do a huge drill right into rock, where he thought there would be lots and lots of biomass from all these bacteria that have lived there over billions of years. They would find oil. And so they spent, I think, $50 million $60 million digging these big holes, and they found a little tiny bit of something, but not enough by any stretch of the imagination to recoup their investment. So that hasn't become a hot topic. It's no longer so hot. So he's become probably more infamous than famous for this. But again, it was an interesting idea. Another very popular idea, at least in the media, is the idea of panspermia, the idea that somehow life came from outside. And you may not realize this, but when Apollo 11, the first mission to the moon, where the first humans on the moon, Neil Armstrong, etc., when they came back, they were actually given biological isolation garments. And when they came out, they were held in quarantine for 3 weeks, because a number of scientists, including very famous people like Joshua Lederman, were worried that in the moon dust would be some kind of microbes. Okay, these microbes would, of course, come on them, and they could be really, they could be some horrible pathogen, and so they wanted to make sure that didn't happen, and so they put these guys in quarantine. So it's not such a crazy idea. In fact, even Lord Kelvin gave a famous speech where he thought that life actually came from outer space. It turns out that bacteria probably could survive in space, particularly if they're inside comets, for quite a long time. And so the idea, which Fred Hoyle, for example, popularized, also Francis Crick, is the idea that life actually came from outer space. That somehow it emerged outer space somewhere, and in a meteorite it came onto Earth, and that seeded life very early, not too long after the late heavy bombardment, and that's how life started. It's an interesting idea. It's not so clear how much evidence there really is for it. So those are all the kind of ideas that there are for how the early proto-life emerged. I hope I made it clear that we don't really know exactly what happened. And in fact, if I'm going to tell you that we can find self-replicating autocatalytic cycles, that's still quite a few steps away from something even as simple as Mycoplasma genitalia with 250 genes, if I'll be very conservative. So the question is, how do we make the next step? And one very popular idea of making the next step is what's called the RNA world. RNA, just to remind you again, so here's this little bacterium I told you about. In the middle is its DNA. In its DNA, it's got a code, basically, what all of its, how it works. So that's its code. It tells you how to make a little bacterium. And the way it does that is there's a molecule called RNA, which comes onto the DNA. The DNA makes a copy onto the RNA, and the RNA moves over to these little guys called the ribosomes. The ribosomes are a little factory. So the RNA is like a little messenger that goes from the DNA to the factory and says, please make me protein X or protein Y. The RNA then goes into the ribosome, where it gets made amino acid. It's all connected together, and out the other end comes a little protein. And there's got lots of these things floating around, these little ribosomes. And so that's what RNA does. So for a long time, people thought, well, RNA is just another messenger molecule. It's not another encoding molecule, just like DNA. And it just acts as a kind of a code from going from one to the other. It's very similar to DNA. It's only one basic chemical which is different. It uses uracil instead of thymine. So that's basically the only difference. And already in 1968, Leslie Orgel and Carl Woese, and apparently Alexander Rich from MIT already claims to have done it 5 years earlier. They already said, well, perhaps RNA has more functions than just passing information from the DNA to the ribosomes. Perhaps it could itself be catalytic, which means it could itself somehow help metabolism and help chemical reactions to happen, which is the job of proteins, basically. And in fact, Thomas Cech and Sidney Altman in the early 1980s, they found exactly this. They found RNAs which are catalytic. So these are RNAs which act to enhance chemical reactions. And that was very, very exciting. In 1986, Walter Gilbert, another Nobel Prize winner, came up with the name or the idea of the word RNA world, which has now become quite a popular thing in textbooks even. And it's popular because it seems to solve the chicken and the egg problem. If it's a catalyst, if it's a ribozyme, actually technically it's called a catalyst, then it can do metabolism, and we know that it can also pass on information. So it can act as a replicator, okay, which is the egg bit, and it can act as metabolism, which is the chicken bit. So it can be the chicken and the egg at the same time. And that's why people got very excited about it. And the basic idea is that it does something like this. So here's a little bit of RNA. It's single-stranded normally rather than double-stranded like DNA, but it can fold into all kinds of complicated shapes. And the idea would be that the very first cells had just RNA by itself. The RNA acted both as a catalyst, so it helped do the metabolism, and it acted as a kind of primitive replicator. It copied itself from cell to cell to cell. Then at some point, the RNA would interact with amino acids, which you know it does, and these amino acids could form little proteins, which are typically much more efficient catalysts. And so over time, it got better and better at making these partial bits of proteins. Till finally you got protein and RNA together. And then at some point, the DNA emerged from the RNA, which was a more stable or a better information storage system. And you got what you have now, which is a DNA which stores the information, the RNA which transfers it from DNA to the ribosomes, and the proteins which are made. One of the arguments for arguments for this might be is that you find, for example, the ribosomes, right? Those things I told you in the previous slide. These little, oops. I hit the wrong button. Yeah, sorry, I got it. Don't worry. There we go. The ribosomes there are made partially of RNA. Okay, so there's bits of RNA which are in them. There's a mixture of RNA and proteins. And the RNA acts as some of the catalysts along the making of the proteins. And since the idea is that ribosomes are very old, the idea is that that's a kind of molecular fossil. The RNA is there as a molecular fossil in the ribosomes, which is where you might expect to find it if you find it anywhere. So that's a kind of indirect evidence, if you wish, for this RNA world idea. Now, there are lots of problems with the RNA world again. One of them is that RNA molecules are very, very fragile. They get hydrolyzed by water. They break up all the time. Their components are very fragile. Nobody really knows how to make some of their components. Even if you have the components, we're not really sure how to form it. And there's also a problem of replication errors, which is RNA doesn't replicate very cleanly because particle is not in this double-stranded system. This means, and roughly speaking, if you have a system, if you've got something which copies itself 1,000 times and you've got a 1 in 1,000 chance of making an error, then if you keep doing that again and again and again, you cross kind of an error threshold, and at the end, you just get your errors multiply, if you wish. And you can more or less calculate how big an RNA you could have to break that error threshold, and it's fairly small, much smaller than what you would need for any kind of primitive life. And here's a very recent paper that just came out where they're looking at this kind of error threshold for ribozymes and for early replicators, and the error threshold for DNA, which is much, much larger. And that's another quite important problem in RNA. So again, this RNA world idea, it was a big surprise that there could be something as an RNA catalyst. It does seem very interesting, the idea that you could have an RNA world, but there are lots of problems, like the fact that RNA is so incredibly unstable. And in fact, there's been a number of alternative ideas that perhaps RNA wasn't the first molecule, but there's one called TNA, there's a few other kind of twists on RNA which are more chemically stable, not quite as good, but almost as good. And the idea is that maybe that one happened first, then RNA came, and then it went through this cycle, which eventually ended up with what we see now, which is RNA, DNA, and proteins. So that's the kind of the next step up. Very popular idea. It's not entirely clear how correct it is. One of the other really interesting things about this, I should make this quote by Joyce. So this Gerald Joyce is a very famous RNA world protagonist, and he says this quite recently. The most reasonable assumption is that life did not start with RNA. The transition to an RNA world, like the origins of life in general, is fraught with uncertainty and is plagued by a lack of experimental data. So that's something from the proponents of RNA worlds. But it's still I think it's a very fascinating idea, very striking. Another thing which I've recently been interested in, which I find one of the things we looked at in this Templeton project, is this idea of the optimality of DNA and RNA. So there's a nice paper here by Freeland and Hurst. Freeland was actually a PhD student. This is PhD work while he was here in Cambridge. It was in Journal of Molecular Evolution, 1998, where you look at, here's, for example, you look at the optimality of the DNA code. And what they mean by that is this. The DNA code works with little sets of 3 codons, and each of the 3 codons codes for 1 amino acid. But there are only 20 amino acids, and there are 64 different codons. So the question is, why are there 64 codons? Well, you can show that if you just use 2 nucleotides, you you can only get 18 different codons. You need to have at least 3. And so initially, people like Crick and others said, well, there's only 20 of those 64 work, and the rest of them are just some kind of garbage. They don't really do anything, or they're just redundant. But it turns out, if you look more carefully at the way the codons code for amino acids, some amino acids have quite a few codons that code for them. Some amino acids have only one codon that codes for them, like tryptophan does. And it turns out that if you can look at the error rates that you get in DNA. So you can have a mutation in DNA where you change a codon from one to another, but it won't have any effect on the proteins, because if it's one of the redundant codons, so one of the multiple codons that codes for the same amino acid, then it just stays the same. And one of the things that Freeland and Hurst did is they looked at which mutation you would have which would be redundant, and then you can make it a little bit further. There's actually quite a few amino acids which can be more or less replaced for each other, and particularly if you look at something called hydrophobicity. I won't tell you what it is, but it's a particular property of amino acids. You don't want to put a non-hydrophobic amino acid where you have a hydrophobic amino acid, and vice versa. So if you do have an error, you would like it, if you do end up having a non-redundant error, you would like that error to at least give you another hydrophobic amino acid. And so they looked at 1 million possible different codes that you can come up with, all different possibilities of redundancy. And then you look at the optimality. So how likely are you to make, if you have a mutation, how likely are you to make an error which changes you from a hydrophobic to a non-hydrophobic amino acid? And it turns out that DNA this is a kind of a bell curve. This is the average susceptibility to genetic error. And this is the code that they found. It's roughly 1 in 1 million. So it's very, very highly optimized. And the question is was that a molecular accident? It just happened to be very well optimized? Or was there some kind of optimization process, some kind of evolution most likely, which in early life allowed DNA to be optimized for error tolerance the way it is? And those are all the kinds of interesting questions that I think are floating around that we'd like to understand and are very, very hard to understand. So in summary, I hope I've shown you that life emerged rather quickly, that there are lots of theories in chemical evolution, and probably lots of speculation would be a better word of saying. There are roughly 2 camps, really, which I haven't really gone into who's in which camp, but there's 2 camps. One is that life is ubiquitous, so that life would emerge really easily again on the Earth. If an asteroid hit us and everything died out, then quite rapidly it would emerge again. And that life may be possible in other solvents. You could have ammonia-based life, etc. And typically astrobiologists, people that are looking for life on other planets, tend to follow these kinds of ideas, partially because it helps get them funding, but partially because there are some hints to say that perhaps this would be true. And the other camp would say that life is a near miracle. It's something that only happened once under very unusual conditions and is very unlikely to happen again. Therefore, if you go to other planets, you're very unlikely to find anything which is alive. And the summary, perhaps I'll take Leslie Orgel again, which I think is a pretty good summary. The problem of the origin of life has much in common with a well-constructed detective story. There is no shortage of clues pointing to the way in which the crime, the contamination of a pristine environment of the early Earth, was committed. On the contrary, there are far too many clues and far too many suspects. It would be hard to find 2 investigators who agree even on the broad outline of events. And I think that's probably a fairly safe conclusion to what people think about the origin of life. This is by somebody who's a big proponent and a very important thinker in this field. So with all that uncertainty, I'll now jump slightly to another question, which is the origin of life and God's agency. And one of the things I think that's happened is that people, particularly religious people, have tended to take this lack of knowledge or this kind of uncertainty about the origin of life and try to put it as an example of God's agency. I was at a talk by a well-known intelligent design expert, Stephen Meyer, also from got a PhD here in Cambridge, on philosophical aspects of the origin of life. And he made the following argument. He said, look, there are 2 ways that life could have emerged. Life could have emerged by chance. I think what he really meant was it happened under very unusual circumstances, or it could have happened by necessity, which I think he meant somebody who believes in the ubiquity of life. And then he says, well, if I use kind of inference to the best explanation, then it's clear that the only way it could have happened was that the designer did it. And in fact, earlier he said this: even if the simplest life owes its origin to an intelligent creator, then perhaps man is not the cosmic orphan that 20th century scientific materialism has taught. So I think this is a very easy thing to say. It's very hard to prove Meyer wrong. It's very hard for Meyer to prove anything right. And the question really is, the question I'm going to talk about now in a kind of switch is, how well does an argument like this work? Particularly, how do people like myself who are scientists react to these kinds of arguments? And before I do that, I need to do a kind of intermezzo about tapestries. Okay, so you're going to wonder, what am I going to say about tapestries? What I'm really going to ask the question is are there different kinds of science? And I want to be very careful here and not say that I think that there are, to first order, the scientific method is the same. We try to make observations, which we then try to explain, which help us reinterpret our observations, and there's a kind of a cycle, hopefully a virtuous cycle, and we understand the world better. And I'm a critical realist. I believe there is a real world out there, and that we're hopefully reaching a better description of it. At the same time, I'm also not trying to kind of solve the demarcation problem, what is science and what isn't science. But I do want to say that I think that there are differences in how scientists look at things, and sometimes these cultural traditions can make it hard for scientists in one culture to understand scientists in another culture. And so I'll give an example, which I'm going to call golemizations of science. And I take this from a very famous book called The Golem by Harry Collins and Trevor Pinch, two quite well-known sociologists of science. This was the first kind of sociology of science, or science technology studies, if you wish, textbook that came out, a popular textbook called The Golem, 1993. And it's quite a nice book. I read it back. In fact, I bought it around that time and read it. And it gives you little case histories of how scientists come up with how they come up with new theories and ideas. And one of the examples they gave, though, which they got criticized quite heavily on, was the question of relativity. This brings us back to our other patent attorney in 1905. But previous to that, there was an experiment, Michelson and Morley, who basically, the idea is that if you have light, okay, then light goes, if there's light is going through a vacuum, then it'll go in the same speed no matter whether you're going towards it or away from it, if light has a constant speed. But if there's some kind of ether, some kind of background, then if you're going against the ether, you'll see a different speed of light than if you go with the ether. So they did a clever little experiment where they pointed things in different directions, and they claimed that light was the same speed in all directions. And if you take up a textbook, your textbook will typically explain to you that this is what Einstein tried to explain. That's how he came up with this idea of special relativity. But in fact, and Pinch and Golem say this: the meaning of an experimental result does not then depend on the care with which it is designed and carried out. Oh, sorry, I skipped a step back. I skipped the slide. Sorry. So Pinch and Collins describe the experiment, and then they point out that in 1933 there was a scientist, a guy called Miller, who did a very careful, who really carefully redid the Michelson-Morley experiment on a much better conditions, better equipment, and he found that there was a difference in which direction you did it. Okay, and so Pinch and Collins say, well, look, even though he published this in 1933, almost 30 years after relativity was first come out, everybody ignored him. And they said, well, they said, so the question was, why did people ignore Miller? Even though he did experiments much better than Michelson and Morley. And why did the community just, why did they not make a big hero out of him, basically? And what they basically says in the Golem is, the meaning of an experiment result does not then depend on the care with which it is designed and carried out. It depends on what people are ready to believe. In other words, the argument was, this is a good example of how the community decides sociologically to declare one thing to be true or the other thing to be true. And I was at Cornell at the time, which is where Pinch was. In fact, he was in the floor office right above mine. And, I'll slow down, thank you. And one of my professors, David Merman, wrote a very famous article called The Golemization of Relativity, where he basically pointed out that the reason why scientists hadn't worried so much about this Miller experiment was because by the time 1933 came around, relativity had been proven in so many different ways that it was a really strong tapestry of many, many different strands which came together. In fact, There are strands like, for example, the fact that you can understand the fine splitting of energy levels on a hydrogen atom from relativity. In fact, even more beautifully, Paul Dirac, just a few streets down here from St. John's College, he showed that you could predict the existence of antimatter by simply taking quantum mechanics and relativity together, that the mathematics proved the existence of antimatter, which was found in 1932, before the Miller experiment. So if you can prove that, if you can predict that there's going to be something like antimatter, which is then found based on relativity, it's very, very hard to suddenly say it can't be true. So there's a very strong tapestry of arguments, which this is how, by and large, people come up with their scientific theories. They're tapestries. They're not, if you can pull on a strand and break it, like you can pull on the Michelson-Morley experiment and break it. But if the tapestry itself is really, really strong, then people don't worry too much about the fact that one or two strands might be broken. And one of the problems with tapestry arguments is that they differ from field to field. So in physics, which is where I was originally trained, you'll find that particularly theoretical physicists put a very heavy emphasis on the unreasonable effectiveness of mathematics, on almost Platonic ways of thinking about out the world. And those have been very fruitful and helpful in predicting the way the world goes. Observation is still very important, but there's a lot of emphasis on theorizing. And the question really is that also true in biology? And I have a little argument here which I'm going to skip because I'm running out of time. It's basically, but I'll come back to it in the end. It's basically an example of a tapestry argument. But I'll skip one back. I wanted to tell a little story. And this is another book by a woman called Evelyn Fox Keller, in a book, Making Sense of Life, which I highly recommend if you're interested in these kinds of topics. She was a theoretical physicist who in the 1950s moved into biology and has been quite a well-known biologist since then. She tells a story about when the first time she taught a bunch of biologists some basic physics, some basic mathematics. And she said she started to use what's called dimensional analysis, which is a very robust idea in the physical sciences. It tells you something about how things relate to each other. And she was telling how this worked, and one of the students raised his hand and said, "Excuse me, ma'am, how do you know that that's true?" And she said, "Well, what do you mean, how do you know it's true?" And she said, "Well, have you done the experiment?" Okay. And this was a kind of an epiphany for her, because she realized that for these students, that kind of theoretical thinking was not very powerful at all. They didn't believe it. They wanted to see the experiment. They wanted to actually see it done. And the reason for that, she argues, is that biology, by its nature, is a lot more surprising, a lot more messy, and the value of pure theoretical thinking in biology is just much, much weaker. And I find myself, as a theorist who works in biology, that a lot of my biological collaborators are very, very suspicious of all of my ideas. Okay? And I think the reason they're suspicious, probably rightly so, is because very, very often these ideas tend to be completely wrong. Okay? Because life surprises us every single time. That doesn't mean that there isn't a lot of internal logic to biology, or there isn't even some kind of theorizing. It's just that the importance of experiment is elevated a lot more. The other thing is that when I talk to my biological colleagues and we discuss experiments, which we're trying to explain, the kinds of things, the way that what they consider to be proven or not proven, I sometimes find the data very noisy, and I think, well, I don't think you've proven anything. But to them, it does tell them a lot. And I think you need to immerse yourself in this. You need to work in this a long time, which is why before you get a good feeling for what you can and can't trust, and essentially what that really is you need to immerse yourself for a long time before you get a good feeling for what really tapestry arguments are in a particular field. And that's not because those arguments are somehow sociologically determined. It's because there's a lot of art, if you wish, to interpreting scientific data, and it has to do with experience, which takes a long time to build up. And that's something which is very, very hard if you're in one field to correctly evaluate in a different field. It's something that you need to really immerse yourself into. And so the argument that Fox Keller makes is that she makes a long argument in this book basically about how these tapestry arguments work in biology and how they change. So I realize I'm running a bit short on time. I think that when you look at something like the origin of life, what you're really looking at is a kind of, use that overused word, emergent science. This is, in other words, things that are much more on the biological side, where new and striking behaviors happen that that are completely unpredictable. Sometimes even in retrospect, a complete surprise. And so people working in those kinds of fields tend to use those kinds of a different kind of tapestry argument than say from the people that work in more physical science fields, which tends to be the types of people that are most likely to critique origin of life work. I'll just give you one example that I'm interested in myself of one of these kinds of surprises. And I'm giving this one because it's a simple one to understand. So this is the question of protein folding. In your body, your proteins are long polymers, like little pieces of string. Okay, when they're made that way, one bit at a time. Okay, and roughly, crudely speaking, the first order, they come out and then they have to fold into some three-dimensional structure. So it's like taking a little ball of string, rolling it around until it makes a little ball of yarn. Now, the question is, how does that work? Okay, how does it fold itself into the particular shape? Because the really amazing thing is that if you take a protein, you can unfold it, and then you can refold it, and it will fold into exactly the same shape, or almost exactly the same shape, every time. Okay? And just to say how amazing that is, if I take a chain of 100 beads, okay, and at each bead I have, say, 10 different ways that I could connect those beads, or 10 different angles they have. In reality, there's of course more than that. Then the first one I can do 10 different times. The second one I can do 10 more times. That's 10 times 10. Third one 10 times, 10 times 10 times 10. So I have 10 to the power 100 different ways of organizing the shape of my polymer to first order. Okay, but what's really surprising is, so you would think that therefore you'll never be able to find the same shape again. Okay, once I've unfolded it, it's got to look through this 10 to the power 100 different configurations, which if you take any kind of normal microscopic speed would take longer than the age of the universe to find its minimum. And yet it finds its minimum again and again. Something called the Leventhal paradox. The question is, how did proteins do that? And in fact, if you didn't actually see them doing it, and I told you that proteins fold this way, then I think most people would agree that argument would completely they'd say I would be a fool, because there's no way that could happen, because there's this many different possibilities. There's no way you could find the same structure again and again. And yet what we see is that it does. And the question is, how does it do that? Well, there's been a lot of work on that, and the argument is basically that there's a little problem with my problem, if you wish, because my problem assumes that I do randomly search through all possible configurations. But I don't randomly search through all possible configurations. In fact, those proteins have a sequence which has not only the final state in it, which is the state which is biologically active, but also the pathway to that state, or the set of pathways to that state, are encoded in it. And in fact, the way a protein works is a little bit like a funnel. That's the language. Like you take a funnel, it's got a little hole in the bottom, which is quite small, constrained, but anything you put in the top will eventually roll around and get to the bottom. It may in fact roll in lots of different ways, but it'll get to the bottom fairly easily without having to explore all possible configurations. And this is something that I think in the last 30 years we've more or less sorted out to first order. There's a whole series of paradoxes like that you can make in biology. If you think about the space of all possible proteins, every single protein is made of amino acids. There's 20 different amino acids you could have at each point. Okay, so if you've got a length 100 protein, which is a really small one, you've got 20 to the power 100 different combinations, which interestingly, if you were to make those, they would use up all the known matter in the universe. Okay, so the question is, how do we find the proteins that we have? This is one of the things I'm working on at the moment with one of my postdocs, and I think we think there's something analogous to funnels in sequence space, which helps us find the proteins that there are. So that's basically, I'm just going to skip this little bit about, this is for later. Yes. So I just want to give you that little bit of flavor about these tapestry arguments, and also about these kinds of, the other section, these kind of surprising arguments, to finish with my last slide, which is this. What about the origin of life? Okay, I hope I convinced you that we don't really know very much about the origin of life at all, and in fact, we may never know. On the other hand, chemical space, which is the space of all possible different chemicals, is so unimaginably huge. There's so many different possibilities that we've only begun to scratch the surface. And again, my colleague Chris has a nice paper about chemical space in Nature 2 years ago. So the problem with this whole origin of life question, and if you want to try to use it as an argument for a proof of God's agency or a proof against God's agency, is that the chemical space is so huge, and moreover that particularly for those of people who are from a biological background are so used to surprises in their tapestry arguments, if you wish, that those kinds of arguments for agency don't tend to weigh very heavily, because people always think, well, you know, how do you know something else couldn't happen? There might be some surprise around the corner. We actually have very little experimental data from what happened to the early Earth. We weren't around. Very little of it is left over. And I think it's quite likely that we'll never know what is the origin of life. And I think I'll end on that.
Right, thanks very much, Art. I'm sure that stimulated plenty of questions. And even though we may never know. Yeah, that's right.
Well, thank you very much. As for the design hypothesis you mentioned Steve Meyer. Actually, as I know, Francis Crick and Wickramasinghe, and even Fred Hoyle and Wickramasinghe, and also Francis Crick were for design argument somehow.
Sorry, where?
They were also making a design argument. Francis Hoyle and Wickramasinghe argued that life should have been created by some aliens. I mean, panspermia was just panspermia by design, not just called
that's right, yeah.
And at some point, Francis Crick argued that life could have been started by humans having traveled back in time, seeding the first cell, which would be their earliest ancestor. Of course, it's a logical absurdity, but faced with the other absurdities and the origin of life, he said that. So this shows that the design hypothesis is really very strong. That people who don't have philosophical, tendencies to accept a design argument, like an atheist, Fred Hoyle, has had to accept that at some point. And it's still marginal, but do you think the general attitude of the scientific establishment might have a role in that? Because you said, for example, astrobiology is emphasized because people need fundings for astrobiology or something. I don't know any major natural science association which will fund the design argument. It's a kind of heresy. So do you think this kind of a cultural approach has a role in the ever-going search for the origin of life, which necessarily, as a rule, excludes the design argument?
Yeah, so I think the difficulty isn't so. I'm sure there's a cultural element to why some scientists would simply be against a design argument out of principle, in the sense of a designer, or even if it wasn't supernatural, it was some space alien. It's not so. These are things that also people like Hoyle and Crick did at the end of their life, which is not always. Lots of people tend to go slightly off base at that time. But I think the real reason why it's hard to make any kind of design argument, except as purely speculation, is simply because we know so little about the origin of life. So if you want to make a falsifiable prediction of some kind, that's very, very hard to do, I think. I think that would be the main reason why perhaps more thoughtful scientists would say, if you're going to make a design hypothesis, the idea that some external intelligence designed the origin of life, you can't prove it for it, you can't prove against it. So what you would need to do is to make something which is concrete and can be tested. And then the difficulty with design arguments is it's very, very hard to say the designer had to do it this way and not that way. Right? So if I say the designer must have made it in this way and then it turns out that it's wrong, I can always say, well, the designer must have done it in that way. That's the difficulty. So, and the problem, so that's really the problem. So when I say something like Meyer, he's welcome to his opinion. I think if I understood him right, he says he can use something like inference to the best explanation to argue for designer. I think that's almost certainly wrong. I just think there's not enough to make an inference about.
Okay, there's hand up in front of you.
Just a naughty point about the culture of science. I was in the same college as Francis Crick when he invented DNA, And he was such an arrogant so-and-so that I used to say that it was purely an artifact in the mind of God. But slightly more seriously, and going back to your tapestries, a rather nice example of that is the infamous suggestion by Fred Hoyle that the chance of life coming to pass by chance was that of a hurricane blowing through a junkyard and producing a jumbo jet.
That's right.
Andrew Huxley, who at that time was president of the Royal Society, answered this and said, effectively, Hoyle had got his sums wrong. In other words, he'd approach it purely from a mathematical point of view. When you looked at it at a more functional, physiological point of view, you were talking about life happening at a particular time rather than over infinite time in a particular place, and much simpler than Hoyle was imagining, and therefore you almost certainly do get life somewhere. Now, I mean, I'm not advocating that approach, but it just shows the difference between them.
That's right. Yeah, that's interesting. Yeah. Okay. I do think just that was a very helpful point. I do think that these like the Hoylean type arguments from large numbers. In fact, this one I told you about sequence space is something called the Hoyle paradox, because he brought this up. They're very. I was hoping to give you with this Leventhal paradox an example of what seems to be a very watertight, hurricane to the junkyard type argument, which nevertheless turns to be wrong in a very deep and surprising and unexpected way. And I think for that reason, biologists are suspicious of these kinds of mathematical deductive arguments, and I think they have good reason for it. They may be wrong, but there haven't really been very many examples of where they have been wrong.
Wouldn't it be the case that the main argument against Hoyle is that the work of scientists is not trying to have some kind of a wind blowing things together, but a very long sequence, each step in the sequence of which has a very high probability, or at least a reasonable probability. I mean, what people like yourself and other origin of life people are doing is not taking a whole bunch of atoms and sort of pushing them together. There's a long sequence of formation of different molecules, first of all, and then more and more complex molecules. And this is a very long and complicated process. I mean, there's no suggestion that what happened was a lot of atoms got together and by an absolutely extraordinary chance, the right combination got together. I mean, that's the wrong metaphor right from the beginning. Wouldn't you agree?
Yes, and no. I think it's the wrong metaphor. I do think still that, and it probably is a long series of things that happened that were quite likely. We just don't have it. Don't have any good example yet of what that sequence was. Yeah. But our expectation is that it's true, that there is that sequence, and that's a philosophical expectation. We could be wrong. We can't prove it either, right to know. So I would rather just say I'm agnostic on it. I think it's very likely that it's the case. I can't personally, even speaking as a Christian, think of why God would have why there would be agency in that particular step but not in something else. So I don't really see a need for it, but I can't say I can prove it. If you wish. I
could just make a quick comment before we pass on around this side and then we'll come to that side. Just as a biochemist, I think a lot of it is also about plausibility, and the more that scientists can generate sort of plausibility structures, then in a sense you tend to go more in the direction in which art is going, in the sense that if you can see how a thing might have happened, then you begin to feel, well, we don't know if that did happen that way, but it sort of makes you believe it somewhat more. And if you want an example about the origin of the genetic code, which we haven't talked about yet, which is always a favorite on these sort of occasions, but there's a nice review last year's annual review of biochemistry about the origin of the code. And one of the ideas which is floating around, it actually comes out of the RNA world, is that the transfer RNAs have dual function. They have to recognize one of the 20 amino acids with one end, and but they also have a codon that plugs into the messenger RNA. They have to recognize the triplet codon with the other end, so that that's what gives you your specificity as you line up on the messenger RNA. So one idea, and that's what the review is about, is that actually the codons were originally selected by the amino acids. Again, it comes back to your chicken and egg, but the idea is it was those recognition, the particular amino acid with a particular transfer RNA, which actually, if you like, got into place the codons which were then carried on. So there's all kinds of plausibility ideas like that, which I think are quite interesting. Now there's a question over here, then we'll go back.
Thank you very much for taking us through the uncertainties in the origin of life. I'm also an ex-physicist who wandered into biology, and one of the things that puzzles me is why at least some of the most influential biology textbooks now begin with exactly the sort of material that you describe as all very uncertain. It's a very strange way for science to proceed. I mean, you begin as a university student, and here's the most speculative material at the front of the book. Any ideas about why that should be?
Well, no, I think that, I mean, that's a very good question. I think if you look in almost all fields, the way material is taught, we almost always use examples that are actually incorrect. And in physics, we use the Millikan experiment. He almost certainly faked it. Michelson Morley. There's many examples that Pasteur probably wasn't entirely clear on how he did experiments, etc. So we do that a lot as a pedagogical tool. I do think something with this origin of life, it is very, if it's taught well, I think in textbooks often, I think they overemphasize the amount of plausibility there. I think they would do better in simply pointing out, here are some, there's like, this quote I have here from Orgel, which is, it's like a well-constructed detective story. There are far too many clues and far too many suspects. And I think if they want to teach well, you want to look at all those clues and look at all those suspects. And I think that's something that could attract young minds into science, because I think there's something very exciting there to be.
I think there's a simpler explanation, which is you have to teach dogma early.
Yes. It could be there's taught as dogma. I mean, I think there's unquestionably a lot of dogma in this field, and there's lots of mixtures of what are essentially philosophy and science mixed together, and that's really unfortunate. I think that doesn't help science.
Okay, there's a question down here, and then we'll get a mic right over there.
Yeah, I was just intrigued about the chirality of nature, and which of these theories, or your own, can shed some light on why amino acids are D and sugars are L?
Well, I don't have a theory about it, and I don't think nobody knows. Yeah.
You talked about the protein folding paradox and the fact that the solution to it was deep and surprising. And it seems to me that doesn't in any sense undermine a design argument. It says that the design is deeper than one might think. So I think one's surprise at the difficulties that people like Hoyle point out is entirely appropriate. But I expect to see a solution which would be deep and surprising and have the same sort of design consequences as the fine-tuning in physics does.
Yeah, that may be true. It's interesting. I mean, I think another important thing about design is that I think probably Michael Roberts made this. This is where I got the story, the analogy from. He points out that we often, human beings, will go to the mountains and we'll see that as something very glorious and beautiful, and it's very normal for people to sense design there. In fact, I was just reading an interview with Francis Collins, the head of the Human Genome Project. Just has a very nice book that's come out, title of which is escaping. The Language of God, that's right. And he talks about his own conversion as a 27-year-old medic and scientist. And he says he was thinking about these things, and he walked out into the mountains, and he walked around the corner, and he saw this beautiful frozen waterfall in the mountain, and he just realized there must be a God. Okay, now we understand the processes by which mountains are formed, the pressure from below, the erosion from above. Okay, but that doesn't take away from the fact that we think that they're designed in some sense, and that they sense glory. That, I think, is the traditional Christian, would be the Christian way of looking at design. So you may look at this idea of the Leventhal paradox and its solution, and the Hoyle paradox and its potential solution, and there may be, you may find it just as beautiful as you would Francis Collins walking around the corner and seeing the mountains.
Explaining something is not explaining it away.
That's right, yeah.
Okay, we'll go here, and then we're all going to get over there.
All proteins has their tertiary stable structure.
That's right. Yeah.
Okay, if we prepare biopolymer from alpha amino acids, which is not exactly natural protein, what about his structure? Will it be again stable?
So if you make it from what, sorry?
From alpha amino acids. From amino acids,
Mm-hmm.
biopolymer, which is not exactly natural one. What about its structure?
That's right. So perhaps if I make sure I understand your question, you're saying if I just take a random sequence of amino acids, how likely is it that it will fold?
Yeah.
Okay, that's actually quite an interesting question that people are looking at the moment. So you can take a random amino acid sequence. You can actually have what fraction of random amino acids sequences are soluble, first of all, okay, and they're linked to that. How much of them are stable? Do they have a folding structure? And it's not known at the moment what fraction that is. Some people say 10, 20, 30%. Some people say a lot less.
They are more flexible?
Yes, and not all proteins, in fact, I oversimplified. In fact, even in your own body, not all proteins are folded. Some of them are naturally unfolded. I'll just point out those ones, the simple ones that fold nicely and don't do complicated things.
Okay, so we'll migrate over that side. Okay, should we go to the back there? And try not to strangle anybody on the way.
I think this discussion on the origin of life can give us some clues about natural history for other living beings.
Okay.
For example, we see here problems of having, for example, 250 genes, of having so much genes. I think from a point of view of Darwinian evolution, let's take, for example, micromutations, for example. What we see, for example, let's take, for example, Archaeopteryx. For example, having a Archaeopteryx wing. For example, having a wing, as Archaeopteryx says, is, I think, needs much more genes than origin of life need. For example, in the many steps of the natural history, we see animals. Many steps between the animals which are presented in the bush of the living beings, I think there are that micro steps even are much more bigger than origin of life. So I think that the discussion of this origin of life are giving us some clues about the entire natural history. Because even with the micro mutations they are made and the animal we see, I think this is much bigger steps than the origin of life. That's all right.
So so to make sure I've understood you correctly, what you're saying is you think that if you look at natural history and the emergence of biological complexity, that those are much more difficult steps to understand than the origin of life is. Is that because you think the origin of life is easy to understand, or are you trying to say that those problems are even more difficult?
No, I think these are in some sense more difficult because, for example, Archaeopteryx was an example. But we have in nature, so we don't have before Archaeopteryx some intermediate forms for wings for Archaeopteryx. But having such a wing as Archaeopteryx, I think, is much more than having 250 genes, for example, again. And for example, when we are making some probability assumptions about the origin of life, we are making it in the entire world. But when we will took it for having such a wing for Archaeopteryx, for example, our probability space is going to DNAs of the living beings. From this space of living beings, we should take such a wing. And so I think to have 250 genes, and we know how sensitive is DNA. Although we don't have having in the beginning chicken and egg problem because we have DNA, and by mutations we will try to explain that kind of having. But I think we will have again explaining of much more than 250 genes because I think that having a wing is much more than it.
So what you're saying, if I understand it right, is that you think this, for example, the formation of a wing on various early dinosaurs is much more complicated than even the 250 minimal genes for a genome. People might argue about whether that's true or not. I think that particular example is not so hard to see how things like wings could emerge, particularly if you're looking at things like evo-devo, which are, I won't get into details of how this works, but there are lots of very interesting proposals about toolbox genes that are highly conserved between almost all known species, and which make things like wings. I think it's Distalis is the one that makes.
Okay, let's just we're running out of time. This question's only on the origin of life. We can't tackle the whole of evolution. we've only got 4 minutes. That's okay, but you know, it's just we're running out of time. So
In 4 minutes, I'm going to ask a cheeky question about something you didn't mention. So, Richard Dawkins had this computer program that he invented to generate sentences. Alistair McGrath pointed out, probably wasn't the first person to point out, that required information already put in. So, this is one of those interesting questions in Origins of Life about DNA and where that information came from and whether it could arise spontaneously or whatever. What do you think about that?
That's right. So you're saying, what about the origin of information? I think the Richard Dawkins example is independent of this to some extent. But people often, it become quite popular, particularly in design, intelligent design literature, to say the origin of information. But I think that is just another way of saying, it's just they're looking at the same question, which is what came first, metabolism or replication? And they're saying the origin of replication is impossible to understand. That's I think that what they're saying is just a fancy way of saying essentially that. And that's, of course, a problem about which scientists argue a lot, about what happened first, replication or metabolism. Some say replication, some say metabolism. And I don't think that actually things like Richard Dawkins' "me thinks it's a weasel" example is actually that relevant to this particular question. You can think of ways in which you can get replication that could be fairly crude replication that could then, through optimization, become more efficient.
Okay, so I think there was Mr. had a question. Okay, so I better make this the last one.
Just to follow with the last thing, you said intelligent design is not a best inference to the best explanation as you think on the origin of life. Do you think it's because it's a bad theology or it's bad science?
Well, do I think intelligent design is not, for the particular question of the origin of life, is it bad theology or bad science? I think the reason why it doesn't follow very well from the inference to the best explanation is because there isn't enough to make an inference from. So you would call that, in that particular case, not very good science, probably. Or perhaps it would be better to say it's not very good philosophy, because it's quite speculative in some sense. It's almost a meta-scientific kind of question. I personally think, and this is something I didn't touch on, but I can afterwards, think that it's theologically somewhat suspect, but that's my own idea.
And if life was indeed designed, what would you expect to be seeing today?
Well, I think this is a very interesting question. Is if life was, I think it's a very interesting question to pose. If there was an external force to the world, and sometimes, even though that language is somewhat suspect, that originated life, what clues would that leave behind that I could observe? Okay, that's a very difficult question.
Because I mean, with that kind of reasoning, it's never ever possible to acknowledge design, and it's always
I think just someone needs to. I myself can't come up with a clear example. I mean, famously, Haldane was once asked, "What would you believe what would it take for you to believe that life appeared instantaneously rather than over time?" And he said, "Well, rabbits in the Cambrian." Okay. So I can make lots of examples of the opposite kind. Because if I can find rabbits in the Cambrian, in the Burgess Shale, together with trilobites, then that would suggest to me that this natural history argument that I have is wrong. So I can think of lots of examples. I can think of lots of those kinds of examples, but nothing that we see suggests that that's the case. And so if it is there, it must be much more subtle. And if there is there, I think it's much more likely, if we're going to find it in biology, it's much more likely we're going to find it in some kind of crude analogies to what we see in fine-tuning in physics, okay, where we're looking not so much at something that we can't explain, but at the nature of the explanation. But I think we're very early on understanding what life is, what it means to be robust, what it means, et cetera, et cetera. I think it's too early to tell.